Dear students, hope you all are doing well. Welcome to another exciting session of the course Mechanical Characterization and Modeling of Tissues. Today we are going to talk about a very interesting topic which is close to my heart, which is on soft tissue simulants. So let's get started. So soft tissue simulants, also sometimes known as the artificial tissues, synthetic tissues, tissue surrogates, these are some of the common names or terminologies used to describe soft tissue simulants. Why do we need such kind of artificial tissues in the first place, right? So one of the key need for artificial tissues is for realistic testing. There is a huge demand of artificial tissues not only in the surgical training market, but as also the ballistic testing and all sorts of, like, armor testing and things like that, right? So typically from a surgical training standpoint, think about a doctor or a medical practitioner who wants to test certain kind of surgical operations or suturing techniques or cutting techniques which they are going to perform in a real surgery beforehand, right? So typically there are some pads on which they can suture. They do not have the mechanical property very similar to the actual tissue. So the idea here is, if there can be a artificial tissue mimic which forms those pads, then you can, as if like you're suturing skin, the real skin, you can suture on those pads innumerable amount of times. Also, if you have a certain model of a liver or a model of a esophagus and different soft tissues or organs which we have already discussed, then they can perform different sorts of surgical training and practices within that, right? Even for diseased and injury cases, right? So these are some of the areas which require a lot of artificial tissues, and that demand is currently not met by the supply, right? The next issue why we require artificial tissues is for because of cadavers and live humans. The testing on them pose a lot of ethical and biosafety issues, right? So ethical and biosafety issues here mean you have to take an ethics approval. You have to make sure when you get a cadaver, you have to take it from a recognized hospital. There has to be certain protocols and norms which are followed, certain good practices, how they have the cadaver has to be stored, how old is that, how the, if you cut a piece of tissue, how it has to be put back, and there are so many norms and regulations around it, right, which gets complex for a new research. And sometimes it's very difficult for researchers or small industries or small companies to really get access to cadavers because they do not have connections with the big hospitals, right? So you cannot be just buying cadaver of a shop, right? So cadavers are difficult to obtain. Also live humans or animal models, live humans of course you cannot test. It's very tricky and highly ethics centric. And also if you're looking at animal models, there are a lot of rules and regulations and laws around animal models, right? You cannot be just taking any animal and run a test on that. So that's not possible. That they have to also go through a certain range of ethical clearances. So having said that, that is one part, which is the ethics part. Second is the biosafety part. Biosafety part is where we are talking about how you're going to dispose the tissue of. Is there any chances of infection which can occur due to this kind of tissue, right? If you if this has been left out, not put in a right freezer in the right temperature for a while, so these kind of issues will occur very often with the experiments and tests which are conducted so far to date, right? However, if you have an artificial tissue which can actually mimic the real tissue, and if you can carry that tissue in your pocket, or if you can just carry it from one place to another without having to think anything about the ethical issues or the biosafety issues, that would be highly valuable for both the surgical and ballistic testing, right? So that's the problem statement which we are trying to address through the set of lectures about soft tissue simulants. That's the background on which we are trying to build up, right? Further into the need for artificial tissues, you need an artificial tissue which at least mimics the material properties similar to the natural tissue. There are a lot of artificial tissues which may mimic the way the structure of a tissue is microscopically, or may mimic how the tissue uh, reacts in terms of different levels of hydration, or how a particular tissue can form a certain block of a tissue engineered material and things like that, or is it similar to an implantable biomaterial. So many of these things are done, but one thing which is often missed out is the mechanical property. You want to make sure when you're making an artificial tissue that, at least for the applications like surgeries, surgical planning and ballistic testing, that it is mechanically similar to the real tissue, right? The second one, not absolutely necessary, maybe necessary for certain applications, which is surface properties. In certain cases where you are looking at whether you're working in the textile industry, for example, you want to make sure you develop textile, or you use textiles to build the clothings which is not very aggressive to the skin. It doesn't cause a lot of friction with the skin, right? So there you need to focus on the surface properties, right? Or you're doing some kind of analysis into things which are getting in contact with the skin, or even implants which are getting in contact with the native tissue. You do not want a lot of frictional interactions that can cause the initiation of certain wounds or infections and non-biocompatibility, right? So sometimes you want to make sure when you build the artificial tissues, based on, depending on the kind of application which you are planning to use it for, you want to make sure that the surface properties are as similar to the natural tissue as possible. Like, I'm going to take an example of the human skin. When we make it out of silicone or some synthetic material versus when we prepare it with hydrogels, it has a nice hydration property, right? The third component, which is extremely important, is the biosafety. So there has been a lot of artificial tissues which are built with different forms of membranes, like the amniotic membrane, like the different silk membrane and other membranes. Many of these may have a biosafety issue. So you want to use materials which are biologically safe, right? And the last thing, which is very, very important specifically for India, is you need low cost and the rightly priced soft tissue simulant, or the artificial tissues which are specific and like which can cater to the needs of our country and several other developing countries where it's a very price sensitive market, right? You want to have something which is much more cheaper than a cadaver and can be easily used, and can be used widely across hospitals and other arenas, right? So having said that, on the right side you can see some of the snapshots of different applications of artificial tissues. The very top, you are looking at a heart model, artificial heart model. The benefit of that is you can not only do surgical practices but also look at certain deformities. What happens if there is a certain kind of deformity in the heart? Can you take the MRI or CT images, and based on that create a model, create an artificial model, and look at if there is a certain tumor or some kind of an ingrowth, how can you extract that and things like that. So a lot of surgical planning, arterial planning, those kind of things can be looked upon. Next is development of sensors or biosensors or wearable sensors specifically. So there are a lot of wearable sensors which will go on top of your skin, or may go inside the native tissue. So those kind of sensors have to be as close as possible in the mechanical properties to the real tissue. That is where you need artificial tissues, right? Because if you have a sensor which is extremely hard, you cannot place it within a piece of tissue. That will cause an adverse reaction or infection. Ballistic testing, as I've already mentioned. So how do bullets penetrate the different layers of skin, muscles, and different organs? And what kind of armors can prevent these different bullet interactions, right? So these kind of things have to be understood in depth, right? And many of these things are missing because there is not a human dummy model which is very similar to the human model. One of the homeworks which you can have is go back and check out some of the literature or research on ballistic testing, and you will see majority of those dummies which you see are extremely non-compliant in terms of mechanical properties. They do not have any similar, they are metallic systems and have no connection to the actual mechanical property of the different soft tissues. They may mimic the mechanical properties of the skull or the bones to a certain extent, but not with the soft tissues. So there is no mechanical mimics of the soft tissues which are available for ballistic testing or blast kind of testing out there in the market, right? The next one is for cosmetic implants or cosmetic surgeries. Planning for that, you need soft tissue simulants or artificial tissues. For building better prosthetics and orthotics, right? So the entire prosthetics and orthotics market needs a lot of improvement, right? You cannot be just placing a metal rod to simulate the natural functioning of a foot, right? If you can make it nice and cozy, where you have the right material properties which are placed along with the like foot mechanism, you can have that as a high comfort wearable prosthetic for the particular people who are wearing it, right? So those things can be looked at. Next is biomechanical testing. You need some soft tissue simulants to make sure there can be some levels of standardization. If I test the skin simulant in India versus I test this in Japan, it should behave very similarly. That is one of the aspects which we have seen while discussing the mechanical properties, is that we taste a piece of skin from here and we are not taking the right location or not reporting the right location or the thickness of the skin, and it shows a property which is completely different from somebody who has tested it in somewhere in Europe, right? So those kind of disconnects are an issue, like with the literature reporting. We need something which is repeatable and reproducible. So that's why artificial tissues and their compositions can uh, help alleviate that uh, issue, right? So then the next one is for personal protection equipment development. So a lot of PPs are worn by the industry, like for industrial functions, as well as we have worn them during the pandemic, and they are worn quite often by the hospital staff. So many of these wearable, like PPEs have to be compliant and do not uh, interact with the skin in a wrong way. That has to be looked upon, right? So in order to do that, you need to have a artificial tissue which is specifically skin in this case, to make sure these PPEs are interacting properly. There are no rashes or long term issues which can come up with these kind of PPEs, right? So this is a broad vision or broad view of the requirement of artificial tissues. Let us get into the depth of the different artificial tissues which have been developed to date. We are going to cover an overview. It is not inclusive of all the articles or all the research ones which have been developed till to date, but like this is still like the state of the art what has been published so far in literature. So let's get started into the different artificial tissue development. Before we take that step, let us just quickly revisit the role of 3D printing or additive manufacturing, which has become a very popular technology in the 21st century, and this is a technology which is widely used for also development of artificial tissue models, right? Specifically for organ development. So the development of complex molds for pouring a certain material and creating a tissue or artificial tissue mimic or an organ, which we have seen for the human brain in one of the previous sessions, needs the intervention of like 3D printing or additive manufacturing. Another thing is you need additive manufacturing for fabrication of fixtures for experimental tests. So like I've also shown during the brain modeling, that the brain not necessarily is to be placed upside down or in a normal kind of sitting posture and being subjected to a compressive testing. That is not the usual case. You want to probably position the brain in different directions, and that has caused a huge variation in the stress strain profile or the stress strain property which we have seen earlier, right? So this correct positioning has to be brought about through development of fixtures which can be very easily developed using 3D printing, right? If you go for the machining route, it's going to take you a long time and probably cost you quite a bit, right? So this is just a showcase of some of the 3D printers, printing assemblies which are there in my lab in IIT Delhi. So we are basically looking at the different 3D printing based products which we want to deliver, mass produce and deliver to several different hospitals during the pandemic. This is of one of the products which was developed, which is of a face shield, right? But the idea here, which I want to highlight, is we are now focused towards the development of the different soft tissue mimics. As you can see on the right side, you're looking at some arterial networks in the B section, and we're also building some molds for generating this test specimens. Like this was designed in SolidWorks, and then finally the red part is the mold, and you're pouring a liquid which is silicon in this case, which has a mechanical property tuned specifically with the human skin, and then you take out the specimens or the samples and you test them all on a UTM, right? So these are the two purposes of using 3D printing. So let us look at the first artificial tissue and the most popular one, which has been under a lot of research for the past 30 to 40 years, which is known as the artificial skin, or sometimes known as the skin simulant, right? So what we did find out, like and this is a while back in 2015 ish, right? We found that no surrogate or artificial skin or tissue simulant exists which can mimic the anisotropic mechanical properties of the human skin. Anisotropic mechanical means isotropic is which has property in the same direction, like every direction has the same property. Skin is highly anisotropic, like most of the other soft tissues, right? So we needed to build some soft tissue simulant which can mimic the anisotropic mechanical properties of the human skin. That was totally missing. That's what we found out from a literature search and a detailed patent search, right? Typically what was used till 2015 or 16, since we developed this artificial skin model, were porcine skin, cowhide, polyurethane gels, or cadavers, right? All these materials are widely used to simulate or mimic skin, and they are used for surgical training, ballistic testing, even to date, right? So one of the key issues with this material can be highlighted or described through this plot or the graph on the left, right? So let's take a look. The real human skin property is highlighted here with a almost like a orangish skin color, which is written as the human skin. So that is the human skin property approximately. Then you have the porcine skin property. So we are looking at the stress strain profile or a stress stretch profile. Stretch is nothing but strain plus 1, just the plot shifted. If you are looking at the porcine skin property, it is much more stiff or stronger than the human skin. So that clearly indicates if we are using a porcine or a pig skin to run some experiments or run some analysis, it is going to give us very different results from what a human skin would give us. That's why it's not a very good candidate. The next one is gels. So gels are extremely soft, like PDMS gels and these kind of gels. These are also often used and called as like an artificial skin or skin simulant in majority of the research articles and other research groups which we have seen. So these gels have mechanical property much lower than that of the human skin. So again, this indicates that they cannot be used to simulate the human skin properties. All right. So with that gap, we started coming out with different sorts of compositions, and we did a lot of hit and trial to find out and determine the right composition of an artificial skin which can get as close to the real skin properties as possible, right? And we finally came out with this black plot, which is the artificial skin, with some error bars showing that we have tested a lot of samples and we have pinpointed on certain compositions which we have tested through a lot of repeatability, at least 50 plus samples on the repeatability standpoint, and finally we could declare it as an artificial skin which is as close as possible to the human skin, and the greatest it has come so far around the world, right? So on the right side, we can look at some of these final products of skin simulants. It typically comes in any form of color, whatever you mix, like with a silicone composition, we call it a polymeric or a silicone based composition. So the blue color is due to a dye. You can have different other colors. The key point here is you are looking at the mechanical composition. So it's a mixture ratio of the hardener and the cross linker. These are the two broad components in a silicon additive cure silicon, addition cure silicon material. So these are mixed in a right fashion, and that is what is patent protected, right? These mixed ratios, right? So that nobody else can use it, and we have the right to use it for making our skin simulants, right? So these can also come in different colors, and this is protected by the US patent number US 100496001B2, known as the biophilic skin simulant. This was filed by me and my supervisor back at the University of Alabama, right? So this work is being continued here in India. This is a screenshot of the patent application on the bottom right, which you can look at, and so these are patents have a different purview. So this is in the purview of the US, and it can be in the purview of other countries as well, and we have a set of patent applications after this in the Indian context, right? So one of the key important parameters which we had in mind while building this skin simulant was to match the human skin properties with a low cost mass producible material. We did not want to pick up a material which is not low cost and which is not mass producible, such as hydrogels. Certain hydrogels are very difficult to process. Whatever materials need a lot of chemical or chemistry kind of operations, they are difficult to mass produce, and sometimes they can be extremely high cost. So we wanted something which is very low cost, suitable for the Indian market. So we came out with the material silicon. The next idea was we wanted to come up with a rationale why we want to develop the artificial skin, and one of the rationale was to study wound suture experiments. We developed this as a part of our research, ongoing research, but the purpose was to study wound suture experiments, which we did not have the liberty of using a cadaver or having an access to a cadaver to try and test those, right, back in the day. And one of the key interest areas out of studying the wound suture experiments was, can we standardize suturing practices? So there is a very nice problem statement here. Think about how the world is moving towards a robotic surgeries, right? Like, so many a times you will see major surgeries in any of the hospitals. You have the option of doing a manual surgery or semi-robotic or a fully robotic. So the world is going to slowly move towards a robotic surgery, right? So a majority of these robots do not have a lot of experience, even though you can say this AI, ML, and all this, words can come in like that they're self-learning, but like computer has to learn from certain inputs, right? So that part is still there. So robot cannot just have a 20 years of experience doing a surgery. That's not possible right now, right? But a surgeon does that. So every surgeon is very different in the market. Like so one surgeon can have a 3 years of experience. That person has a very different ways of conducting a surgery or suturing, and another person has a 20 plus years of experience. That person has a very different way of doing a surgery. So which surgical practice should I translate or take and input it into the robot? So you may argue that let us go with the 20 years one, right? But they may not be well versed with the latest technologies or the latest happenings out there. So which one is the right practice? So in order to do that, we did a lot of wound suture experiments with the standard practices, and we wanted to standardize some of the suturing practices so that a person who is basically 3 years into the system, or who is let's say 1 year within the system and is a medical practitioner or a medical trainee, they have a standard method of suturing, as well as this can be also translated to a robot, right, which can of course self learn, but as a starting point can get the right suturing practices. So this was one of the key ideas while developing the artificial skin, right? So we wanted to determine suture forces for surgeries, which are very important for a robot or any standard practice to understand that how much suture force has to be applied once you have placed a suture, and determine the suture forces for robotic surgery. So these were the key problem statements which we were following. So every invention, this is an invention, every discovery, every development comes with certain backgrounds. Like, we do not start one day saying let me build an artificial skin. How would I know what is being done so far in literature, right? So everything comes with certain background, some problem which we are trying to solve, like some healthcare problem which we are trying to solve, and then we focus and see what is missing. So we found that an artificial skin which is highly reliable and similar to the human skin was missing. Can we develop that, right? So that was the idea, and that is how we started into our voyage in developing the artificial skin. Next, we wanted to also look at the training on suturing practices for different wound sizes and shape. So what happens when you have a different wound size? Is there any difference, right? If you have a small wound versus a long wound versus a broad wound, what is the difference? We wanted to do some parametric study to understand what is really going on with the suturing practices there. Do they have to be sutured in the same way? If we can learn this, we can feed this information also to the surgeons as well as to the robots. So having this kind of background in mind, we developed this artificial skin simulant, right? So what you're looking at on the right top is a set of skin samples, human skin samples, which we have tested first. So that is step one of development of any artificial tissue or tissue simulants, is you look at the real tissue, right? Either you find out that information of the test results from literature, where they have clearly declared what is the strain rate, what is the sample size, what is the size, different dimensions of the specimen, how many specimens were tested. These kind of information has to be there. That is number one technique. The other technique is, if you have access to that real skin or real tissue through a cadaver or something, you test that first and set up some benchmarks that these are the possible stress strain profile or the properties which I have to match. So unless you know where you have to go, you will not be able to go there. So you set a goal there. So that is what we did first. We tested on real human skin, and we found and we took the specimen sizes and everything, and we determined all the stress strain profiles due to that, right? And next, we developed different compositions of artificial skin, and then we wanted to validate by doing some suturing experiments here and on the real skin and see if the experimental results matched, right? This is an extra validation step. Besides checking whether the material properties match, whether besides the matching of the material property, does it behave similarly when we do a similar operation like suturing? That is also must. Because let's say the material property is almost similar, but when you are trying to suture, it's a very highly elastic or hard material and the skin is a very soft material. Then it doesn't solve that purpose, right? So the overall purpose has to be solved through different levels of validation. So as we started our journey into the synthetic artificial skin development, we went through different stages of mixing and matching different compositions through hit and trial. So what we did, and the most of the key results are captured within this large plot on the very left. So you're looking at a set of test compositions. Here we have mixed four different parts of silicon. They come in two different set of additive cure silicon. One type was of a certain shore hardness, where based on the ratio by weight of the hardener and the crosslinker, it will throw a certain property within a certain range. And again with the second set, it would be the same. But when we mix all these four components together, then you mix the two sets, and then you finally get a 4-part mixture ratio, which you can very fine tune to get certain properties which are unique, right? So having started with that, we got a range of properties from the very beginning, very soft to very hard. So most of these plots which you're looking at from the very bottom to the top went like that. We kept on changing the compositions and looking at what is the property. We changed the composition, created 5 samples, tested it on the UTM and measured the properties, right? And we placed all of these properties together in a single plot to compare properly whether the property is changing So for example, we had started with PDMS, we changed the PDMS compositions 4-5 times, it still yielded the same property. So we realized that PDMS is not the right material to develop skin, right? So the next one was silicon. We have used polyurethane. Polyurethane was way too hard. Silicon was somewhere in between, where there were a lot of options, and we started building that different compositions of silicon. So as we did that, we also compared each of the stress strain property of the different compositions with the real skin properties. What we also realized from the literature as well as from our tests on the cadaveric skin, that skin has not a single stress strain property but has a range of properties depending on the location of the skin, whether I am taking a piece of skin from the forehead or from my arm or from the thigh, it has a different property because the thicknesses are also different. Also there is a difference in the property due to the anisotropy or the Langer lines, how you are cutting a sample, whether I am cutting a sample parallel to the Langer line or vertical to the Langer lines, right? So these kind of location and anisotropy had a huge effect on the skin properties. So we basically defined a clear range of from where to where property in the stress strain plot can be called a material as skin. Okay, that needed to be defined. So now if you see on the plot, we have defined something which is known as the skin lower bound, and then we have another one which is known as the skin upper bound, right? So within the lower and upper bound, if any of our compositions lie, then we can safely say that this is some composition of skin or some form of skin. That is what we did exactly. We kept on building the different compositions, and then we mixed and matched that this 4 or 5 compositions are within the range of real skin properties. Then the next step was fine tuning that, right? We had to develop one specimen, at least one composition, or pick up one composition, which we are calling it as 90-10 control specimen. Control specimen is something which you use for repeatability and reproducibility tests, right? So if I say, so what is repeatability and reproducibility, I'll explain both these terms because these are very important for experimental tests on soft tissues, right? But the 90-10 control specimen specifically refers to a composition of 90% of one set of silicon and 10% by weight of another set of silicon. Then within that silicon sets, they are distributed like 50-50% by weight or something like that, right? So that was the 90-10 composition naming. So what is repeatability? Repeatability is when I test the same sample. So let's say I have the same sample which I have tested 5 times. Does it show the same property? It can never show the same property. It shows a little bit similar property and probably some variation, but the variation should not be drastic. So if I see a plot like this, the other plot should not be like this. It should be somewhere close to that with maybe a 3 to 5% variation. Those kind of things are okay, right? That is the repeatability test. I want to make sure that whether this same sample gives the same stress strain profile when I test this 5 times. The next one is repeatability by composition. When I develop the same composition using the same set of silicons 4 or 5 times, does it lead to the same sample composition which I test and gives me the same property? Otherwise, what I would say is the composition is very sensitive, or if something changes by a little bit, the entire property is going to change. That should not be the case, right? We need a composition which is reliable. So if I test this composition tomorrow versus I build some samples today, they should behave similarly in terms of stress strain profile. So these are the two types of repeatability here. Next is reproducibility. Reproducibility is I am using some type of silicon which is available in India and a set of silicons, and I build this particular composition, let's say this 90-10 control composition in India, and I test it 5 times, it is giving me a certain stress strain profile, right? The same composition is now known by somebody in US, let's say. So they purchase a silicon from their end, some other company, and they create the same composition with the right hardness of the silicon and everything, and they test it on their machine. Let's say the results are completely off. That means the results are not reproducible. Is it fault of the research group in India? No, not really. So we have to clearly declare what machine was used, what was the strain rate, and what was the exact company. So if somebody around the world is not using that same configuration, same machine, and same brand of silicon, it may give a little bit slightly different result. But if it is completely off result, then that's not a reliable or a reproducible composition, right? So we did a lot of reproducibility tests to look at different silicons and make compositions with that different brands of silicon, and to make sure that they will give us the same property every time, right? So this is repeatability and reproducibility I've explained. So what we did next was we took the 9010 control specimen, and we found out that it is closer to the skin lower bound, so we could consider it as very close to the skin lower bound. And then we repeated the test around 30 times, right? So we got a lot of stress strain plots for that, but they almost matched, and we could draw an error bar within 5%, right? So that meant that we had not only created an artificial skin which lies within the range of lower to upper bound, but we had created a repeatable and reproducible artificial skin, which is of importance, right? So these are some of the terms which we should learn while we are trying to develop soft tissue simulants. We ran a set of other tests to make sure that the soft tissue simulant or the artificial tissue which we had built for skin is as close to possible to the real human skin properties. So what were those tests? Repeatability tests I have already explained on the very top right, control 9010 test specimens. Second is similar low and high strain modulus. We wanted to draw a low strain modulus by dropping a tangent from starting from the 0 point in the plot to the curve, as well as we did the same from the top of the plot to the curve. A tangent, we dropped a tangent. Those are known as low and high strain modulus in terms of slope, right? So that gives us an idea till what limit a particular skin simulant material is going to stretch to, and let us compare whether that is similar to the real skin. Third was ultimate tensile stress. What is the maximum limit to which you can pull this, or maximum amount of load which you can apply to this skin specimen before it completely fractures or ruptures? Can we compare that with real skin properties, right? So what we did see here on the figure on the right is we saw a certain fracture pattern, and the silicon based specimen did fracture. But what we did notice was this fracture pattern was very different from the real skin fracture, how it would fracture, right? So that means it's not a very reliable metric to study ultimate tensile stress properties, and also the ultimate tensile stress values were way different. They were within certain ranges, but they were still very different. So that's why we could very clearly state that we have created a repeatable skin simulant, reproducible skin simulant lying within the stress strain nonlinear profile, but we cannot particularly say that the ultimate tensile strength or the fracturing of this tissue simulant model is going to be similar to the real skin. So that's a clear caveat or an assumption which has to be declared, right? So what are the shortcomings and limitations of our work that those were declared, right? So this is the synthetic artificial skin introduction, how we had done this. Based on this, we moved a little bit more advanced. We next went to something which is known as a hydrogel based artificial skin, right? This uses a lot of knowledge about the biochemistry, which we are not an expert in, but we adapted some of the biochemistry standard practices based on the literature. So here we are using porous hydrogels with feel similar to the natural tissues in order to create the artificial skin. Hydrogel compositions were optimized to obtain the skin mechanical properties. So what happens is hydrogels typically have around 90% plus water contents. That's why they are called hydrogels, and rest is the gelation powder which mixes with the water to create those gels, right? So you have a very small play area there. 5 to 10% is what you can change the powder, and rest is water, right? So when you use a lot of water, the hydrogel becomes very soft. When you use less of water, the hydrogen becomes a little hard, but you have very little room to change the composition. So changing the composition as well as to have some mechanical stiffness so that it doesn't break, as well as having a mechanical property similar to this skin, was a very challenging task. We had to spend a lot of time changing different compositions, like very specific chemistry had to be looked upon to create those hydrogel based synthetic skin. So here is an example on the left where you're looking at the stress strain profile. You're looking at the human skin properties, which is in the dashed black color. Then you have one chemical set of chemicals which were mixed to create an hydrogel, which are known as PNI-PAM slash AA2, right? But that produced properties which were much greater or stiffer than the actual human skin. Then when we change this to PNI-PM slash GO2, right, graphic oxide, that again created a property which was much lower than the human skin. However, when we mixed PNI, PAM, AA2, GO2 all together in the right mixed ratios, which are the right chemistry, then we got a property very similar to the human skin, right? But one thing to note here, the overall property of this material was much lower than what we have observed previously. Here you're looking at a stress of uh, 20 megapascals or 25 megapascals, and here you're just looking at a 200 kilopascals, which is fairly low. So you are playing in a small range. If you remember, most of the in vivo techniques, like which has indentation, imaging, it gives you the force displacement or stress strain in a very small range or with small loads. That's the same case with this hydrogels, right? We could not test them or operate them at a very high load. They would disintegrate or break down, right? So, but still this was a nice find, and we could create skin simulants which we could subject to compression as well as tension like in a universal testing machine, as you can see on the figures on the left. The top one is a compression test, right one is a middle one is a tensile test. We did a cost calculation because we were really focused not only on the material but also how this would function in the Indian context. So we did a cost breakup analysis. So we saw was around 10 by 10 centimeters, like this is the size of the skin. So 10 by 10 centimeters of skin with this or a artificial skin developed with hydrogels with a close to 0.4 centimeters thickness. This amounts to 40 cc of hydrogel with a density of 0.1 to 1 gram per cc, which will lead to approximately 40 grams maximum mass. So the cost breakup of the major raw materials like poly N isopropyl acrylamide, which is poly N PAM, and so this was then PNI PM is 0.1 gram, would cost around rupees 29, right? Polyacrylic acid, which is the PAA, this would cost for 1 gram around rupees 4. Graphene oxide GO would cost around rupees 6 every 0.2 grams. So and the distilled water is the cheapest. 34.5 gram was needed to build one sample, and that would cost rupees 5, right? So other chemicals and supplies were costing around rupees 3. So the total cost of material for a 40 cc hydrogel based artificial skin was around rupees 47, right? So with this 40 cc of hydrogels, it by comparing the densities, it was almost equivalent to 93.2 grams of silicon, right, in terms of mass and volume, right? So in terms of mass specifically, so with a density of silicon of 2.33 grams per cc, what we saw was a hydrogel will cost at the amounts of rupees 500 per kg equivalent of silicon. So per kg of silicon, equivalent of silicon, the hydrogel will cost around 500 rupees, which is not a lot, right, which is a reasonable amount. So we did find that this hydrogel is cost effective, But what was an issue was to develop all these compositions, and with our limited knowledge of biochemistry, we could not make the hydrogels very consistently or repeatably with a high reproducibility or repeatability. That was not possible, right? So from a mass manufacturing standpoint, this seemed a little challenging, which may be overcome with some collaborations in the future, right? So this was one of our attempts in the hydrogel based artificial skin development. The next interesting soft tissue simulant which we developed was of an artificial artery. So the human artery, which I may have explained in the previous sessions, is a very complex systems in terms of the tissue. It has three layers of the tissue, commonly known as the intima, which is the innermost layer, followed by the media and the adventitia, which is the outermost layer. Each of this layer has a different composition of the tissue in terms of properties as well as the direction of fibers. In one of these the fibers are coiled, in one of these the fibers are longitudinal. So properties of each layer are very different, and it has a unique structure, and arteries have undulations and structure changing which goes from one point to another in the human body, right? So another part was this is highly patient specific, so not all the arteries look the same or with the same thickness or the same undulations across human bodies, right? Like, so that's why we realize that this has to be a patient specific approach in order to develop an artificial artery. So we did a patient specific 3-layer human artery model, and we could create it as close as possible to the real artery. What was the background or the purpose? We are going to revisit this purpose statement again and again whenever we talk about any artificial tissue development. So the purpose here was to look at stents. So whenever there is a blocked artery, right, blocked coronary artery, specifically coronary artery is the main artery in the human heart. So whenever there is a blocked artery, there is an angioplasty procedure which is used to open it using a stent, right? So how do you size a stent? How do you know the right size of a stent which needs to go inside this artery, which already has a clot, right? So this is typically taken through imaging, 2D imaging or a two-dimensional imaging technique. It is shown. But what may happen in 3D may be very different. The stent may have a diameter from the imaging standpoint of this much, which is, let's say, 3 millimeters or 4 millimeters, but going in the other direction there may be a lot of clot where the stent may get completely stuck, or may produce or generate a lot of pressure on the arterial wall, which may cause other difficulties with infections as well as the blood flow, right? So what we realized was, first thing, we wanted to look at the artery from a model standpoint in 3 dimension, and second is we want to have a mechanism where the right size stent can be selected for any surgery, right? It should not be oversized, because oversized stents can damage the wall of the artery and create thrombosis and further infection, and an undersized artery will not be able to serve its purpose of opening up the artery properly to remove the clot, right, and allow the passage of blood. So we wanted to make sure we could develop patient specific artificial artery models before it goes for the stenting operation. A medical practitioner or a doctor is able to insert a stent within this model and try and test what should be the right stent size and stent type which should go within this system before doing the surgery. So that was, it's known as a pre-surgical planning as an overview, right? So this was the purpose behind creating the patient specific 3-layer human artery surrogates or the artificial arteries. The fabrication method was fairly similar in terms of the materials, but in terms of the coiling of the artery, or it has a undulating surface, we had to use 3D printing, right? So the first step what we did was we took a set of MRI images, like from one of the patients. We created an arterial model, right, like a coronary artery model, and sectioned it in the place where there was a blockage or something like that, right? Like we basically sectioned this particular area where the stent has to be placed, or which we have to model here. And what we got as a model after stacking it using a DICOM file and image segmentation is the hollow part of that artery, which is known as the arterial lumen, right? So we 3D printed the arterial lumen shape or structure. The next step is we wanted to grow the different arterial layers on top of this lumen and take this and in the top of the lumen 3D printed model, and take this lumen model out so that there is a proper hollow within the arterial system. So after the MRI 3D printing, what we did was we created the 4-part silicon mixture in different ratios. Each of these, so now we had 3 different ratios simulating each of the properties for the different layers. If you look at the graph on the right, what you can see here is basically you are looking at a very cluttered plot. Let me just go into that. Maybe I can zoom in. अच्छा, इधर चला गया। अच्छा, इस पे यूज होगा ना। ठीक है, थैंक यू। मैं इधर से कंटिन्यू करता हूँ, वो थोड़ा प्रॉब्लम है। अच्छा, ये इधर चला गया। ठीक है, तो यहाँ से स्टार्ट करते हैं। ठीक, so what you're looking at here is the plot on the right, where you are seeing a lot of very cluttered information. What this information really tells us, and I could not use the entire screen to present it, it shows the values of the mechanical properties or the stress strain profiles which has been collected in literature through study of the artery. So there have been researchers, specifically there is a researcher known as Holzapfel in Europe. He has worked extensively in the areas of soft tissue, and he has developed different measurement techniques to measure the properties of the different layers of the arteries. So he has been able to dissect the different layers and measure the properties, right? So these properties are what you see on the top right of the plot, and then most of the straight looking lines are the compositions of the silicon which we have used to closely simulate these properties which have been derived from the literature, right? But as you can see, majority of the curved lines could not be matched very well with the linear lines, right, which we are seeing, and we are looking at kilopascals. That's why majority of our compositions are looking very linear. If you go with a higher load, it will kind of bend over, right? So even though the curves were not very nicely simulated, we could still at a very low strain kind of safely say that we had some compositions which we could identify, which could properly simulate the mechanical properties of the intima, media and adventitia. So this is what we did as an exercise with a lot of different material compositions, testing them again and again, looking at the ranges and looking at which particular composition is very close to this particular profile of stress and strain, which has been derived from literature testing on real arterial tissues, right? Then we developed a very new technique. Once we had developed the compositions, we developed a very new technique which is known as a time control dipping technique for layer thickness control. So another very important thing here with the arteries are what is the thickness of each of these layers. I cannot randomly put it as 1 millimeter or 2 millimeters. They are all properly documented in literature. Those are the layer thicknesses which we found out, and what we came up with a technique where we have a certain composition of silicon and we wanted to dip this 3D printed lumen model within this after a certain time, because within that time the silicon has cured a little bit, let's say 20 minutes, and it has hardened a little bit. After that hardening, it is going to create a layer which is of, let's say, 1 millimeter. If I pour this particular model into the silicon within the first 5 or 10 minutes, it may create a layer which is of 0.2 millimeters, for example. So one of my job for a few months was to do this time control dipping all through the day to figure out after what time should you dip to get this much amount of thickness which I desire. That thickness is very clear from the literature, right? So after doing all this exercise, I could come up with the most perfect artery model. This is what you're seeing on the very right bottom. So this is an arterial model with a clear intima media and adventitia, exactly the same layer thickness what it should be in a real coronary artery, and the mechanical properties are also very similar. Before doing this, we came up with some single layer arterial models as well as a practice, which is shown in the middle picture, but the right bottom picture is the final product which we came up with. Now this model could be tested with stents and other different medical devices, right? So this was our development of an artificial artery. The next model which I am going to talk about is of a artificial brain tissue. So artificial brain tissue, I had already explained in one of the previous sessions. We found out there were no simulants to date which can mimic the mechanical properties of the brain tissue. So we first of all created some silicon based compositions to make brain tissues tensile specimens, which has tensile properties or pulling properties which are similar to the brain tissue. We came out with different compositions, tested them on the UTM at particular strain rates, which has been clearly stated in literature. So we derived the values from the literature. Then this part I have already explained in previous sessions, where we have created an artificial brain model, and basically within this brain model we have not only created the structure based on like the actual human body designs and the human body dimensions of a patient, as well as we have created a whole brain model, and using additive manufacturing we could make two parts, and uh, using the compressive property, picking up the right mixture of silicon, we made this model, and finally we got them tested in three different configurations. The top portion of the parietal lobe, prefrontal cortex of the frontal lobe, and the temporal lobe. These were the three different configurations in which we see impacts typically in a human body, and these are the uh, three examples which we took, like in order to do the mechanical characterization in different strain rates. And finally we could compare the results. I will just get to the key highlights here. So what we did find out was, due to the orientation change, there was a huge difference in the mechanical property which was reported, but with the strain rate the brain tissue did not see a huge change in the mechanical property, right? We also were able to curve fit everything. When we went from load versus displacement to stress versus strain, almost those differences also vanished. We saw a very unique, almost single property of the brain tissue, irrespective of the direction in which we placed, and irrespective of the strain rates, right? So as a conclusion, what we found out was, for the brain tissue artificial model which we had developed, it had a very consistent stress strain profile, and stress strain profile is what we should consider for testing or comparing any soft tissue as we go along. And based on the strain rate and changing in orientation, specifically the brain tissue did not behave very differently, but the other tissues can, which we are going to cover next and talk about more. So stay tuned. We are going to finish it here for this lecture, and we will pick it up from here and talk about more soft tissue simulants. Thank you.